Buenas tardes a todos. Antes de que se ponga más fuerte la lluvia, vamos a dar inicio a la plática del Dr. Robert Chalkley. So, Robert, thank you very much for being here. He's an associate adjunct professor at the Mass Spectrometry Facility and the Department of Pharmaceutical Chemistry at the University of California, San Francisco. Robert has an undergraduate degree in biochemistry from the University of Sussex, and since then he has been working with mass spectrometry. During his time as an undergrad student, he was already working with mass spectrometry, and he combined mass spectrometry and analytical chemistry at SmithKline Beecham, which later became GlaxoSmithKline. And he also got a PhD. He was telling me that his degree was granted by the University College London, but he did his research at the Ludwig Institute for Cancer Research, which I recommend you take a look because it is a very interesting organization that has research centers in many places in the world. So since then, he was working with developing mass spectrometric methods for the study of the regulatory modification of O-gluconacylation. And he is working at the National Mass Spectrometry Facility of the University of California, San Francisco, first as a postdoc and now as faculty, and he has developed several methods for the analysis of glycoprotein. He has recently chaired the Proteome Informatics Research Group of the Association of Biomolecular Resource Facilities, which performs studies to assess the research community's abilities to analyze proteomic databases. So thank you, Robert, very much. And without more delay, I'll give you the floor.
Yeah, well, thank you for the opportunity to present here. If you have any problems understanding me, just let me know, and you'll have to get used to my accent, I'm afraid. So I've been asked to give a talk on glycoconjugate analysis using mass spectrometry. Practically all of the work I do is analyzing proteins and peptides, so there's going to be a very heavy emphasis on glycopeptide analysis, but I will briefly mention other types of glycoconjugates. So with genome sequencing, this allows you to predict a protein sequence, but we don't normally analyze intact proteins using mass spectrometry because it's very difficult. Other problems are the sequence in the database may be wrong by one amino acid, say, and that will change the mass such that we can't identify what it is based on the mass, or the sequence in the database may be a pre-protein with a signal peptide on, so the protein may become processed, and so the final product is not the sequence that's in the database, or the protein may become post-translationally modified. So a modification may be added to the protein, which is also going to shift the mass. And so obviously that's the emphasis of what I'm going to be talking about today, where modifications such as glycosylation are going to change the mass of this protein. And so what we normally do is we digest the protein into peptides, so shorter pieces, and then we analyze these peptides and try and, based on the sequence of the peptide, go back and work out what protein we were starting with. And so we use two different types of fragmentation that Cesar talked about at lunchtime if you're in his seminar. One is called collision-induced dissociation, which is very good for analyzing the sugar part, and then we use another method called electron transfer dissociation, which is very good at analyzing the peptide part. So mass spectrometry, we're measuring masses, and so we can analyze modifications because a modification corresponds to a mass change. So if we were analyzing acetylation, the mass would increase by 42 Daltons. If we were analyzing phosphorylation, the mass would increase by 80 Daltons. So obviously for glycosylation, there isn't a mass for glycosylation because there's a lot of different modifications. But for example, if the modification is a hexanac, then the mass would increase by 203 Daltons. And so, yeah, if in a position where we're expecting a serine which has a mass of 87 Daltons, instead we see something that has 290 Daltons, that's 87 plus the 203, then we can say there is a hexagonal attached to that serine. And so here we have a fragmentation spectrum, and we have a mass 87 for serine, 99 for valine, and then we have a gap here of 290, which corresponds to the serine with the hexagonal attached. So based on this, we're able to identify that there is a hexagonal modification on this serine. And so in this particular example, it's very easy to identify the peptide sequence. So we're reading backwards. So it's S, V, S with the hexagonal attached, LGAA, LGAA. And so we're able to identify the sequence of the peptide and the site of modification. So in glycans, there are a number of different sugar building blocks. There are pentoses, so 5 carbon units, hexoses with 6 carbons, hexosamines, N-acetylhexosamines, deoxyhexoses, and uronic acids and sialic acids. So each of these types of building block, there are a number of different versions of them, but if it's a pentose, it's always going to have a mass of 132. If it's a hexose, it's always 162. So using mass spectrometry, we can, based on the mass shift, we can identify the type of sugar residue, but we can't necessarily tell you what particular isomer. So it's difficult to differentiate between a galactose and a glucose because they have exactly the same mass. So here is a figure showing the different types of glycoconjugates you might find in a eukaryotic cell. So starting at the top, we have proteoglycans. So these are attached to particular proteins, and they're very long sugar structures. Then we have more conventional glycoproteins, so N-linked glycosylation and O-linked glycosylation through serines and threonines here. Then you can have glycosphingolipids, so glycolipids, where the sugar is attached to a lipid. And then these GPI-anchored glycoproteins, so glycophosphatidyl'inositol, where you have an anchor between a lipid and a protein, and the bridging part is a sugar. And then the final type of modification here is this O-gluconylation, which Laura mentioned I've done quite a lot of work on analysing, but I'm not going to say a great deal about today. So very briefly, mass spectrometry analysis of different types of glycoconjugates, glycolipids. So that there are two main types of glycolipids you may come across, glycosphingolipids and glycoglycerolipids. In both cases, the sugar part is relatively similar, but it just differs by the lipid that's attached. And so, in the case of these glycosphingolipids, they actually represent the majority of the glycoconjugates you'll find, say, in the brain. So there are a lot of these around, and glycoglycerolipids are structurally fairly similar. They have a different lipid component, and they're more common in plants than they are in animals. So glycolipid analysis. So for mass spectrometry, we have to analyze charged components. The lipid part is generally very neutrally charged, and so most of the properties of the molecule in terms of charging it come from the sugar component. So in some cases, the sugars are relatively neutrally charged, or if you have a sialic acid group, this is going to give a negatively charged group to your molecule. So for this reason, sialylated glycolipids are normally analysed in the negative ion mode. So instead of looking for a plus proton, we're looking for a minus proton, because these are more likely to lose a proton than they are add one. The MSMS analysis of glycolipids is relatively similar to analysing free glycans, because the lipid component is very stable and won't fragment, whereas the sugar component fragments much more easily. So the fragmentation you'll get will largely tell you information about the sugar component. Glycophosphatidyl'inositol anchored proteins. So these are proteins that are anchored to the membrane through this particular linkage. So it's attached through the C-terminus of the protein. You can somewhat predict which proteins are going to have this type of modification based on the sequence, but it's not very reliable. The way these types of proteins are normally analyzed is that there are enzymes, phospholipase C and phospholipase D, that can cleave the GPI anchor off of the lipid. And so what people try and do is to identify modified proteins, they try to find the C-terminal peptide from the protein that will still have this sugar modification attached to it. So why are we analyzing glycopeptides? Glycanalysis is much easier to do. If you're interested in analyzing N-glycans, it is much simpler to enrich for the glycans, because you can treat your sample with an enzyme, PNJS, that is going to cleave off all the N-link modifications. And so it's very easy to produce a pool of N-link glycans. The fragmentation analysis is simpler because you've only got a sugar component. You haven't got a sugar component and a peptide component, which behave differently. And glycan analysis is still the best way if you want to determine linkage information. As I said, limitations of mass spectrometry, we're just measuring masses, so it's difficult to determine between different isomers, and it's also very difficult to determine linkage, because if we have a sugar structure and we knock a sugar off, it's very difficult to tell which residue, which sugar linkage that corresponded to. But the weakness of glycan analysis is that you lose all information about the site-specific, and if you're analysing a mixture, protein-specific glycosylation. So only by analysing the glycopeptides can you say this sugar structure is on this serine, this sugar structure is on this asparagine. And so this provides complementary information to the glycan analysis. And so one would hope that people would be interested in doing both glycopeptide analysis and glycan analysis if they wanted to comprehensively analyse a particular protein or particular sample. So glycopeptides can be categorised by how they are linked to a protein. So we have N-linked glycosylation, where most of it is through an asparagine residue. You can have O-linked glycosylation, where it's through hydroxyl groups. And then less common types, you can get C-glycosylation. So this is a carbon linkage of glycosylation that can occur on tryptophans. And you can get phosphate-linked glycosylation. And this glypiation is the GPI anchor that we mentioned earlier. So I'm mainly going to focus on N-linked and O-linked. So the CID behavior of a modification depends on the type of linkage. So in this particular example here, this is an N-linked glycopeptide, and when we fragment this particular peptide, the sugar stays attached to the asparagine, and so we get this shift of 317, 114 of which is from the asparagine, and 203, if you remember before, is the mass of the hexac. And so here we can just read off the sequence, and the mass of this shift says this particular asparagine has a hexac attached. However, if I show you a spectrum of an O-linked glycopeptide, you can see, although the mass of the intact peptide is 203 Daltons higher, so we know there is a glycosylation on there. When we look at the series of fragment ions, we see that all of them correspond to the masses of an unmodified residue. So what is happening here is that when we put energy into this molecule to fragment it, the first thing that happens is it cleaves the glycosidic link such that you get the sugar residue here, which is this 203 plus a proton, because we're measuring charged molecules, and the mass of the peptide with the sugar having fallen off. So from this, we can identify the sequence of the peptide, but we have no information to say where the linkage was, which serine was attached. I mean, I'm cheating giving you the answer, but based on this data, you wouldn't know which serine has the modification attached. So here's the same CID spectrum. If I fragment the same peptide with this other fragmentation method, this electron transfer dissociation, which instead of breaking the weakest bonds, fragments using a free radical mechanism. So it breaks at the same place, irrespective of the strength of the bonds. So here you can see now, instead of getting a mass of 87 for the serine here, we've now got a mass of 290. So the sugar is staying attached to the serine that allows us to identify the site of modification. And so this electron transfer dissociation was a real step forward in O-linked glycopeptide analysis, because it allows us for the first time to reliably identify sites of modification. So here is our peptide. From now onwards, I'm going to be labeling spectra with the types of fragments on them, so I'm going to remind you of the nomenclature that's being used here. So that there are 3 different types of peptide backbone that could be cleaved. When we're doing this collision-induced dissociation, we're mainly going to cleave the blue bonds here, and if it's the N-terminal part, we're going to call it a B ion, and if it's the C-terminal part, we're going to call it a Y ion. And Y3 means it's the C-terminal part and it contains 3 amino acids. Conversely, if we do this electron transfer dissociation, we're going to be cleaving the next bond along. So instead, we're going to have C ions for the N-terminus and Z ions for the C-terminus. So, O-gluconacylation. So this, in many ways, is different from all other types of glycosylation, in that this is addition of a single sugar residue, N-acetylglucosamine, onto serines and threonines of nuclear and cytoplasmic proteins. So this is occurring inside the cell. This isn't on secreted proteins or membrane proteins. This is occurring inside the cell. It's found in all multicellular organisms. So it's very widespread. And it's different because it's inside the cell, but also because it's a transient modification. So there is an enzyme responsible for adding this, an O-gluconac transferase, and an enzyme responsible for removing it, an O-gluconacase. And so this modification could be put on and put off in response to signaling. The modification is involved in cellular response to nutrition, but also a lot of other stresses. And it's been particularly well linked to diabetes and Alzheimer's disease. And it's also elevated in cancer. So this is a regulatory modification that is affecting the activity of the protein. For example, in Alzheimer's disease, the link is that the tau protein, which is one of the major proteins involved in causing Alzheimer's, this protein becomes insoluble rod-like structures. In these insoluble structures, the protein is heavily phosphorylated. However, the O-gluconac occurs on similar residues, and so when the protein is O-gluconacylated, it prevents these tau fibrils from forming. And so there is limited evidence to say that through O-gluconacylation you can actually, in rats, you can reverse the effects of Alzheimer's. So, as I said, O-gluconac analysis is an O-link modification, and the ability to analyze this was transformed by the availability of this electron transfer dissociation. So prior to the availability of ETD, there were roughly 20 known sites of modification. So there are antibodies against them, and so they could say, well, this protein is modified, but they didn't have a way to identify the site of modification. The method they would be using was pick a residue, mutate it, and see if the modification disappears using the antibody. So, but following the availability of these methods, so the first study, we identified 58 sites in the one study, and in a more recent one, we've now identified 1,750 sites in a single analysis. And so we've gone from 20 in 20 years' work to 1,750 in a week's work. And so the increase was mainly due to this electron transfer dissociation, but it's also mass spectrometers have become faster, so they can acquire more data in a given period of time. And it's also because we've developed improved enrichment strategies, so we're able to produce a sample that is heavily enriched in modified peptides and then we can then analyze them. And so O-gluconic analysis is about as simple as you're going to get in terms of glycopeptide analysis, because it's just a single sugar residue. So the major challenge is purely site localization. So this is a particular example where we're confident that this is the peptide sequence, but we're struggling to determine whether the modification is on this serine here or the following serine. And we're labeling C and Z ions. This is ETD data. And what this is actually showing, it's probably not easy to see. So those labeled in blue are unique to this assignment, and those labeled in green are unique to this assignment. And we have this Y10 in blue here, but we also have a Y10 in green here. And so what is actually happening here probably is this is actually a mixture spectrum of the two different versions of this peptide with modifications on each site. And so this is a fairly common problem in the O-gluconic analysis, in that we're doing reverse phase chromatography to try and separate components, but these two peptides, the same peptide with just a different site of modification, co-elute. And so we're getting mixture spectra. So collagen is a major structural protein. It's a major part of the extracellular matrix. And it forms these very rigid triple helical structures. So many of the lysines in collagen become hydroxylated. So a hydroxyl group is added onto the lysine, and then this hydroxylated lysine can become glycosylated. And so there's two types of glycosylation. You either get a galactose or glucose galactose. The function of this glycosylation is largely unknown, but the pattern of glycosylation does differ depending on the tissue. So it must have some functional relevance. One potential effect it might be having is affecting collagen turnover. Is the glycosylated collagen more stable? And one piece of interesting information to support this idea is some data from where they're studying collagen from this 120,000-year-old bison fossil. So this is dinosaur fossil, and that they're able to extract collagen from the bones of this. And doing an analysis, they found that there was a very heavy enrichment of glycosylated peptides. So that they found a very large number of this particular peptide from collagen with the glucose galactose attached. And so would a protein normally be stable after 120,000 years if the sugar wasn't on there? Normally, I would think of a sugar as being relatively unstable, but it's very surprising that on such an old fossil as this, you're finding large amounts of glycosylation. Proteoglycans, so these are another type of O-linked glycosylation, and these consist of proteins with very long repeating unit sugar structures on them. They're a major component of the extracellular matrix, but they're almost never analyzed as glycopeptides. It's nearly always release the glycan and then analyze the glycan, because these sugar structures are much bigger than the protein itself. And so most of the emphasis on this is analyzing the glycans themselves. So the examples I've given you so far, we've just had a single hexnet attached, which is obviously very simple. In reality, you have much bigger sugar structures. So here is a peptide that has a high manocyte structure on it. And so the characteristic ions we have in here, we have the mass of the intact peptide with one of the hexnacs attached. So as I told you earlier, the bond between the asparagine and the first hexnac is stable, so that's not going to fragment. But in CID, all of these other glycosidic linkages can fragment. And so you get this characteristic ion of the peptide with the one sugar attached. And then in the low mass region here, we have fragments that correspond to sugar residues. So the 204 here is a hexac. The 163 here is a hexose. And then we have a 366 here, so 204, 203 plus 163. So this is hexac hex. And so we're getting ions in the low mass region that tell us this is a glycopeptide. And we have this characteristic ion up here that says this is the mass of the peptide with one hexac attached. And then we have other fragments in here, and basically all these other fragments are the intact peptide with different numbers of sugars attached. So this peak here is the intact peptide with 2 hexacs attached, then hexac, hexac, hex, hexac, hexac, hex, hex, and so on. And these are also this is one. There is one peptide cleavage. The Y15 is cleaving between alanine and proline, and then we're getting another series of ions with the sugars attached. So the CID of this N-glycopeptide is giving us a fair amount of information about the sugar structure, but the only information we have about the peptide is the mass based on this Y1 ion. So the bottom spectrum here is a CID spectrum of a different peptide. So this particular peptide here has a mass of 3,503 daltons. And this bottom one here has a mass of 2,693 daltons. So this is nearly 1,000 daltons smaller. However, if you look at the fragmentation spectrum, they look almost identical. And the reason for that is that the only difference between the two of them is the sugar structure. It's the same peptide, but with different sugar structures on them. And because the fragmentation is mainly just of the sugar side chain, you can very easily recognize and say, well, this peptide is probably the same peptide as this one, whatever the peptide sequence is. I don't have very much information on what the peptide sequence is, but these two are clearly related to each other, so they're probably the same peptide with different sugar structures on them. And by comparing the two masses of these and knowing what the masses of all the different types of sugar residues could be, you can come up with a composition for the difference between the two of these. So here is a corresponding ETD in glycopeptide spectra, and so this is very different. And so now we're getting lots of peptide backbone cleavage, and we've got a very big shift here between this Z11 here and Z12 here, which corresponds to the mass of the asparagine with the whole sugar structure attached. So here, the sugar is staying intact in the fragmentation, and we're able to see the side modification and all the whole composition of the sugar. But we don't have any structural information about the sugar other than the mass. And here is the corresponding ETD spectrum of the other peptide, and you can see here that the fragmentation is a little bit different now because we're getting peptide backbone cleavage with the sugar still attached, and so that means the masses of these fragments are now changing because this C6 here contains the first 6 amino acids are IAP, A, S, N, and it now has this sugar mass attached, whereas before it had a much bigger sugar attached. And so the masses of the fragments are now changing. So olleic glycopeptides, as I said before, these are labile under collision-induced dissociation, so we're going to see mainly D-glycosylated fragments. So in this particular example, the characteristic ion is what we're calling the Y0 here. So this is the mass of the intact peptide with no sugar attached. And then in the low mass region, we've got different sugar fragment ions. So 204 is our hexac. We've come across that one before. This 274 here comes from a cyanic acid. And we have a 366, which we've seen before, so that's hexac hex. And so we have fragments from the peptide backbone that don't have the sugar attached, and we have some fragments to say what sugar residues were attached, but we can't say what the site of modification was. And so here we have another CID spectrum of a different precursor. So this one weighs 3,451, whereas this one weighed 3,102. And you can see that the fragmentation spectrum look almost identical because, again, all the sugar residues are falling off. And so basically the only fragments we're seeing are telling us information about the peptide sequence, which is the same. And so the slight difference in this one is we've now gained another peak here at 316.1, and this is an acetylated sialic acid. So sialic acid already has an acetyl group on it, but it can become further acetylated. So here's an extreme example. So these are the low mass regions from 8 different glycopeptide spectra, and in each case it's actually exactly the same peptide, but we have a different sugar structure on each one. So here we've got a GalNAc-Gal sialic acid. Here the sialic acid has become acetylated. Here we've got 2 sialic acids attached. Here we've got an N-acetyl neuraminic acid and an N-glycolon neuraminic acid. So this is a different type of sialic acid residue, which isn't expressed in humans. This happens to come from mouse, and so you get an extra level of complication because mouse uses another type of sugar residue. And then we've got other versions where the acetylations are in different positions. So this is interesting in that in this particular one we see a fragment that's hexnac and the acetylated sialic acids. So based on this, we're able to say that the acetylated residue is attached to the GalNAc and not to the galactose. So occasionally you can get some structural information. So here is a table of these sort of low mass fragment ions that we're getting that can give us diagnostic information about types of sugar structures that are attached. So I've gone through most of these before. So having got these diagnostic ions, you can actually make use of them to try to find glycopeptide spectra. So if all glycopeptide spectra produce this 204 ion from the hexnac, then there is software available. So this program is called MS Filter. It's part of a package called Protein Prospector that we developed. And what we can do is we can take a list of fragmentation spectra and say, I only want those spectra that contain a 204.08 in the fragmentation spectrum. And so you can give it a file of everything you fragmented and tell it to come back and give me only those spectra that contain this particular fragment ion. And you can go further steps in this. You could say, well, it's got to contain 2 of these fragment ions. And so you can go through and say, well, I only want those that contain the hexnac, but also a sialic acid. And so you can then target which spectra you want to analyze based on the presence of these ions. And the presence of these ions can also be used to more intelligently acquire the data to start with. So, as I said, most glycopeptide CID spectra produce this hexnac-oxonium ion, this 204. And so what you can do is you can tell the mass spectrometer to only acquire ETD spectra if the CID spectrum produces this 204. So it can go through, fragment everything it sees, and then when it finds something that's producing this 204 ion, so we think it's a glycopeptide, only then acquire the ETD spectrum. So why not acquire ETD all the time? If ETD is much more useful for glycopeptide analysis in general. The CID is much faster, so you can analyze more components and screen much more quickly to identify potential glycopeptide precursors. The CID data is useful in that, it's telling us this is definitely a glycopeptide, which is useful information, and it can give us some information about the sugar composition based on these low mass ions. And the CID data is much more useful if you've identified what the peptide sequence is. If you've got these 2 spectra that look very similar, and so you're sure that they're the same peptide, and you've identified the sequence from one of them, you can infer the sequence from the other, and the CID data is more useful for telling you information about the sugar composition. So another useful feature is that glycopeptides have different neutrally charged glycans co-elute. So in most of this analysis, we're doing liquid chromatography interfaced directly to the mass spectrometer. So the peptides are being separated based on hydrophobicity, hydrophilicity, and if they have neutral charge, they're going to elute at almost the same time. So here we have extracted ion chromatograms. So these are like, if it was an HPLC, this is like a UV chromatogram. This is telling us when a particular component is eluted. And you can see we've got 1, 2, 3. We've got 7 different versions of this same peptide with different sugar structures attached. A lot of high mannose, and there's a complex one here. And you can see that, and this is time on the x-axis, and so at 49 minutes into the run, all of these peptides are eluting at nearly the same time. So what this means is, if you identify a particular glycopeptide at this particular time, you could go back to the mass spectrum and look for other glycosylated versions of this same peptide, because they're probably eluting at the same time. So you could look at the masses and say, well, there's another component in here that's eluting that is 163 Daltons higher in 162 Daltons higher in mass. So that's probably the same peptide with an extra mannose on there. Conversely, if the sugar structure is charged, then it's going to change the elution. So sialic acid, the negatively charged group, is going to change the properties of the peptides such that they're not going to co-elute. So this is the same example I showed earlier, where it's a single peptide with these 8 different O-glycosylations on here, and you can see that the retention times are all spread out based on the different sialic acid compositions. So how do we enrich for glycopeptides? So, obviously, if we're digesting a protein, the majority of the peptides are not going to be glycosylated. And so if we want to come up with methods to get rid of the unmodified peptides so that we can focus on the glycopeptides. So one of the most popular methods to use is hydrophilic interaction chromatography. So, and the reason this is used is that the glycan is very hydrophilic. So glycopeptides are going to be more heavily retained in this hydrophilic interaction chromatography than the unmodified peptides. So you can bind your glycopeptides to this column, wash off the unmodified, and then elute the glycopeptides. Another type of approach is to use lectins. So lectins are sugar-binding proteins, and there are a number of these different lectins, and different lectins have different sugar specificity. So some of them have specificity for sialic acid, some of them have specificity for galactose or for glucanac. And so by using these lectins, you can enrich subpopulations of glycopeptides. So I mentioned at the bottom here, wheat germ agglutinin. If you read the literature, it's supposed to have specificity for terminal glucanac residues and sialic acid residues. However, I'll show you some data in a few slides that shows this basically enriches everything. And so we've been using this as a way to enrich for glycopeptides. The third method I mentioned on here, immobilized metal affinity chromatography. So this is a method that's very heavily used when people are trying to analyze phosphorylation. So the phosphate group has a big negative charge, and so it binds to these immobilized metals. However, sialic acids also have quite a big negative charge, and so they are also enriched in the same way using these metal affinity chromatography. And I actually published a paper fairly recently where I took someone's phosphopeptide data set and showed, well, here's some glycopeptides in your data that you didn't analyze, because they didn't look for glycopeptides. So how does this lectin weak affinity chromatography that I mentioned I've been doing work? So weak germ agglutinin, as I said, it officially has affinity for glucanac and sialic acids. The affinity for a single glucanac is very low. So the weak germ agglutinin has 4 binding sites. And so for a complex glycan where you might have multiple terminal glucanacs, you can bind very tightly and you can do a bind and elute strategy. And so what we were trying to work at is we were trying to study this O-glucanac where we had a single sugar modification, and we were trying to work, well, can we use this weak germ agglutinin to enrich for these O-glucanac modified peptides? And quite a few people tried this, and they weren't having any success doing a bind and then elute strategy. So what we did was we switched to a chromatography method where the glucanac modified peptides are going to elute later. So there's no bind and elute, but we have this big broad peak eluting, and we found that the tail end of this peak is enriched with glucanac modified peptides. And so using this, we identified a bunch of O-glucanac modification sites in a couple of different papers. And so this was working reasonably well, but still in this tail, about 90% of the peptides were unmodified. So it's not a great enrichment strategy. So what we then worked out, is that if we take our first chromatography, if we take that tail end and we then reload it onto the column and run it again, then that tail is much more enriched in modified peptides. And if we take that tail and load it on for a third time, we go from, after the first step, it's 5% glycopeptides, to after the third step, greater than 80% of the peptides in there are now glycopeptides. And so using this, we've come up with studies where we identified 1,700 O-glucanac modified peptides from mouse synapse. And then these 2 other papers where, analyzing this tail, not only was it enriched in O-glucanac, it was enriched in all types of glycosylation, both N-linked and O-linked, and independent of whether there was a terminal glucanac or a sialic acid on there. And so we were getting a very broad spectrum of glycopeptides. And so, well, this is my sort of depiction of why this is actually working. So if we have two distributions where the blue is the unmodified peptides and the red is the modified peptides, you can see here the blue is dominating this region. But as you go through these steps, even though the chromatography is not changing for the modified peptides, the amount of the unmodified is going down, so the proportion of the modified is increasing. So that's how we create data. Now we've got to work out how to analyze it. Identification of your glycopeptides using software and database searching is not easy. Part of it is many researchers only have collision-induced dissociation data. So they don't have the ETD data that is most powerful. And so one way to get around this is they use a protocol here where they enrich for the glycopeptides. Most of these have been using this Hillick chromatography to enrich. Then what they do is they split their sample in half, and one half of it they remove the N-glycans using PNJZ. So this is going to convert their N-linked glycopeptides into unmodified peptides, but where the asparagine has been converted to an aspartic acid. And so there is a 1 Dalton mass shift that is diagnostic to say this peptide was previously glycosylated. And so they analyze the deglycosylated sample to come up with a list of peptides that they think were formerly glycosylated. So having got a list of peptides that in their sample that they used to be glycosylated, they can then analyze their intact glycopeptides and only consider as potential modified peptides those that they identified from the deglycosylation step. So that they have from this step here, they have a list of masses that correspond to glycosylated peptides. From here, they have a list of masses that correspond to glycosylated peptides. And so that they're trying to look for mass differences between the deglycosylated and the glycosylated that correspond to sugar structures. and based on that, they're going to try and identify glycopeptides. And so, obviously, one weakness of this approach is you're only going to identify N-glycopeptides. But there have been a couple of studies that have done this that have identified a few hundred sites using this approach. So how do we do that? So this is another program in this protein prospector software that we develop. And so we're going to be analyzing ETD data to try to identify glycopeptides. And so what we're going to do is instead of telling it a list of modifications that we want it to look for, we're going to say we want to look for any modification between mass of 100 and 3,000 that can occur on asparagine, serine, or threonine. So we're going to treat the sugar modification as just a big mass modification of some mass, and we're going to try and identify peptides where, based on the fragmentation, that's telling us the peptide sequence, and then on one of these particular sites, we've got this big mass modification. So what does that actually look like in terms of results? So here it's identifying a peptide, and it's got a mass modification rate. So it's saying this particular fragmentation spectrum, if there's a mass modification of 1,216 on the asparagine, then it would match this particular peptide. And so it's all the same peptide with different mass modifications. Some of them are duplicate spectra. But we've got a 1216, we've got a 1378, we've got a 1540, we've got a 1702, and we've got an 1864. If you're quick with math, you'll be able to see that all of these differ by 162, and so these are all different glycosylations with different numbers of high manos on there. And so from this big search, we can plot a histogram of these mass modifications that are observed. And so we've got a big peak here at 203, which corresponds to a hexnac, and then we've got a whole bunch of other different masses in here, most of which correspond to different types of glycosylation. Next slide, yes. So those labelled in light blue correspond to N-linked glycan compositions. Those labelled in red correspond to O-linked glycosylation compositions, and those labelled in purple could be either N-linked or O-linked. And so nearly every peak in here is explained as a glycan modification. So how do we work out what 1241 or 1540 corresponds to? So there are several tools available that can do the calculation for you. So I'm giving you one example here, which is called Glycomass Calculator. The website's here, and so you can tell it what sugar composition you want to consider, and it will go through and do the calculation for you.